My name is John Mandelman. I am Chief Scientist here at the England Aquarium, Vice President of our Anderson Cabot Center for Ocean Life, which is the organization's Applied Ocean Science Research Institute, which is fantastic that we have that as part of our New England Aquarium. The aquarium is a nonprofit conservation organization. We've protected and cared for the ocean, its animals and its habitats for over 50 years. We were founded in 1969. Our speakers this evening are renowned shark biologist, Dr. Greg Skomal, and writer Ret Talbot. They're longtime friends of our aquarium and share our passion for protecting the blue planet. Personally, it's been a pleasure to not only know Greg, but collaborate with him, and fortunate enough to call him a friend for many years. And I just wanted to digress with a quick little anecdote, if I may. I think it was about 1999 or 2000. I had just moved to Boston. It was before I started grad school at Northeastern University. And I had the courage to get in touch with Greg. Now, for those that know Greg, slightly, not always up on the technology end of things, maybe lagging a little bit behind. He had a flip phone until I think about 6 months ago. But Greg, however I got in touch, I think it was email, was kind enough to meet with me. I had no formal marine science background. I didn't have a degree. I barely was capable of speaking in this language, but he still met with me. And he took the time. And I remember, I think your office was down on the vineyard at that point. And I went over to the vineyard and met with Greg, and he took the time and gave me such great advice about my career, grad school. And in all the years since, as we become friends and collaborated on papers and projects, and now some of the team and our Anderson Cabot Center here at the aquarium collaborate with Greg, and even a couple of them were mentioned in the book, which we're going to learn about tonight. He just always gives incredible time to people, to students, to colleagues, to collaborators. Nobody's questions or attention to want to talk to him is ever a waste of time for him. It's always very warm and inviting. And I can't think of a time when I've worked with Greg or hosted a lecture where he's spoken and somebody hasn't come up to him with that same, hey, can I talk to you? I'm a student at this university, or I'm thinking about doing this with my career. And I just find it so inspiring that he always has the time for those people. And I think a lot of people, similar to one of the icons that I think, based on the book, is one of his heroes, Matt Hooper, in the novel and ultimately the movie Jaws. I think that that character inspired Greg, inspired many other marine scientists, including myself. Greg has inspired many others to follow in his footsteps, myself included. So he's a hero in that light, and that he's driven a lot of us towards ocean conservation. I just wanted to mention that going in. So now back to the program, our regularly scheduled program. So Greg is here tonight, along with journalist Ret Talbot, to discuss their recently published book, Chasing Shadows: My Life Tracking the Great White Shark, a story the Publishers Weekly says offers readers an intimate look into the sharks to the white shark's life from the safety of dry land. Greg is an accomplished marine biologist, underwater explorer, photographer, and author, and leading shark expert, not just white sharks, but shark expert in the Atlantic and around the globe. He's very, very renowned for his work. His day job is that he's a senior fisheries biologist with the Massachusetts Division of Marine Fisheries and the director of the Massachusetts Shark Research Program. He's also an adjunct professor at the University of Massachusetts in the Intercampus Marine Science Graduate Program, an adjunct scientist at Woods Hole Oceanographic Institution in Woods Hole, and a member of the Explorers Club and Boston Sea Rovers. Some of you may know him from Sea Rovers. He has authored dozens of scientific papers and appeared in several films and television documentaries, including programs for Nat Geo, the Discovery Channel, PBS, and numerous television networks. He's also regular on Shark Week and Shark Fest, and is the author of the Shark Handbook. Greg holds a master's degree from the University of Rhode Island and a doctorate from Boston University. We are also joined this evening by award-winning freelance journalist and photographer Ret Talbot, whose work focuses largely on marine ecosystems and fisheries issues at the intersection of science and sustainability. His work can be found in publications such as Nat Geo, Discover, and Yale Environment 360, among many others. As a science writer, Ret has traveled the world with marine scientists to cover their work in the field, including places like Papua New Guinea, Sulawesi, and Belize, and is frequently working closely with scientists like Greg to bring compelling stories about science to a general audience. Ret is a graduate of the writing programs at both Wheaton College and the University of St. Andrews in Scotland. And as I just learned, has a really interesting span of moving around in this world, including Wyoming, Scotland, born in Connecticut. Both these gentlemen, by the way, were born in Connecticut. So they are New Englanders, but a little bit south. But without further ado, it's my honor and pleasure to welcome Greg and Ret to the stage. And following their remarks, we're going to do some Q&A that will invite questions from you all. And I should have said, welcome to the virtual audience as well. And those of us joining remotely, you will too have a chance to ask questions after the formal remarks tonight. Welcome to Ret and Greg.
Thank you, John. I appreciate that. Growing up, I was always told that even I was from Southern Connecticut, as was Greg, and that's not New England. It's got to be North Mystic to be New England. But thank you. I appreciate the New England status. Before we get going, Greg and I really wanted to thank Maria and Mark, who did such an extraordinary job helping to coordinate this event, making us feel very welcome here. I have white shark glasses for each of you that were designed by scientific illustrator Ret Talbot, who did the illustrations and the maps in the book, and also happens to be my wife. So these are for you. We'll give those to you later. And for John and the New England Aquarium, I have a set of poor beagle shark and white shark glasses for you, and just really wanted to thank you for the work that you and the aquarium do for our blue planet. And as everyone in this audience knows, this work is so important, more so now than ever. And so thank you very much for everything you do. So if we can give a round of applause to the aquarium. So our book is a conservation success story. It's a conservation success story about the resurgence of the white shark population in the Western North Atlantic. It's a story that is told through the eyes of Greg Skomal, who is a shark biologist who quite unexpectedly, as you'll learn tonight, found himself in the middle of this resurgence in the white shark population along the New England coast. But it was very important to us early on that we wanted to make sure that the shark remained at the center of this story. And so we begin the book and we begin each subsequent chapter with the shark. Chapter 1. The morning sun cut through the green-hued water, casting bright downshafts of light and that undulated along the slate gray back of the 11-foot shark. It moved effortlessly, or so it appeared, but also with inimitable purpose. If a fish could swagger, this is surely how it would look. From the side, its pointed snout, large dorsal fin, and crescent-shaped tail disclose its identity, but it's the color, the abrupt demarcation between dark upper and glaring white underside that is unmistakable to even the most casual observer. The white shark is commonly called the great white because the mere mention of its name conjures up larger-than-life imagery. In the face of those jaws or that dorsal fin, those are, of course, the most common tropes. Anything short of hyperbole falls flat. The shark's confidence becomes arrogance, its efficiency, aggression. It's frequently called mythical, and while it does occupy a central role in the mythologies of many cultures, there really is no need to idealize or embellish. The actuality is enough. The hard reality is that this animal is about as close to perfection as any animal in existence today. Everything about the white shark is purpose-built to excel in one of the Earth's most challenging environments, as it has done for millions of years. The white shark swam here when glacial Lake Cape Cod was still draining, when humans emerged, and when the dinosaurs went extinct. The white shark's ancestors managed to survive every mass extinction since they arrived on the scene more than 450 million years ago. To call the white shark an evolutionary success story would be like saying Leonardo da Vinci was a reasonably competent artist. The white shark is no myth. It is no accident or aberration. It is the result of a sequence of precise adaptations achieved over millions of years, and it has culminated in this, a near perfect, albeit little understood animal, swimming steadily north along the outer Cape on a mid-September morning.
That right there is the magic of Ret Talbot, because he did such a phenomenal. Our partnership was fantastic because, you know, he took my story and the biology of the white shark and helped us put it together in something that was fun to read. You know, this isn't a textbook. You know, it's a little bit about me. It's the trajectory of my life, and chasing shadows is almost as much about, you know, chasing dreams. And but it's the story of the white shark and our quest as scientists, not just me, the scientific community, to learn about this animal. John, thank you for the incredible introduction. I remember that day in my office, and look how far you've come. It's unbelievable. You know, I think of you as an amazing scientist and a great friend, and it's a real pleasure to be here tonight and be introduced by you. But this whole story, you know, for me, you know, so I'm going to talk about me a little bit. I don't want this to be a Greg Skomal story, but I think it's a great representation of how quickly we ramped up and began learning about the white shark, you know. So that's me as a really little kid, you know, and I'm one of 7. And as John said, and Ret, I'm from Southern Connecticut. I'm no longer a Yankee fan, but I did grow up a Yankee fan. You know, I grew up the way the crow flies, probably 5 miles from Long Island Sound, but Long Island Sound in the 60s and 70s wasn't the most pristine body of water. So much of my love of the ocean came from watching this guy on television. This is Jacques Cousteau. Anyone here remember who that is? He's a pioneer in scuba diving and bringing the ocean into our living rooms. And he did that for me. It started in black and white and then evolved to color. In 1975, I saw a movie named Jaws. And at this time, I had already established a number of aquariums in my home, and much to the chagrin of my wife, my mother, and I was really fascinated by the ocean, by fish. And then Jaws came out, and I became fascinated by this movie. You know, in 1975, I graduated from middle school. So you could start doing the math and realize just how old I am. But that character in the film, played by Richard Dreyfuss, is what inspired me. And as John indicated, not just me, a whole bunch of people. We did a film a number of years ago called How Jaws Changed the World. And it wasn't necessarily about how it scared the hell out of people. It was about it and how it inspired scientists and filmmakers and divers and such. So I decided at a pretty early age that Matt Hooper had a really cool job and a really cool boat. And I thought it would be really fun to be a shark biologist. So let's fast forward 4 years. 1979, I graduated from high school. Okay. That same year, a scientist by the name of Frank Carey, who was at Woods Hole Oceanographic Institution, did a pioneering study where he built one of the first electronic tags and put it on a white shark and followed it for 3 days. And so this is a picture of Frank. I got to meet Frank, you know, before he died, and I got to work with him a little bit. But he published this paper, Temperature and Activities of the White Shark. And that came out a couple years after he did the research. He was the very first person to track in real time the movements of this animal in 3-dimensional space. This was technology that was just starting to emerge. It's called acoustic technology. I'll talk a little bit about it tonight. This isn't a science-based talk as much as it's hopefully an inspirational talk. But Frank was not only recognized here in the Atlantic as a great scientist, but on a global scale, because nobody had ever done this before. To get a 3-day glimpse into the biology of this animal was unheard of, let alone using this new technology, which was an acoustic tag. And the way acoustic tags work, they emit a very high frequency sound, and you follow that sound with the boat. If you put a transducer into that acoustic transmitter, you can get the depth of the shark. So you could see what it's doing in 3-dimensional space. And then you can put in a temperature sensor on it. And these were all really primitive back then. You know, this is the 70s. And you can get what temperature it's at as well. And he did all this. And it was absolutely amazing. And I think the scientific community recognizes it as one of the first papers to get a glimpse into the biology of this animal, not only in the Western North Atlantic, but in the world. But that is the only study that persisted here in the Atlantic relative to what we know about this species in terms of its three-dimensional movements and its ecology. So all that was going on while I was in high school. I graduated from high school, moved on to the University of Rhode Island. And one of the things I did at URI is I started working very closely with a professor at URI. Now, he studied really small fish called sticklebacks. I didn't give a damn about sticklebacks, but he was a really good scientist, and he taught me a bit about the scientific method, and I took that as a stepping stone. But he also told me about a job opportunity working for a program down at the Narragansett National Marine Fisheries Lab. That program was headed up by a gentleman named Jack Casey. And Jack Casey, although I did not know it at the time, was another pioneer in shark research. And he worked very, very closely with Frank Carey at Woods Hole Oceanographic Institution. And Frank had been studying sharks since the early 60s. And Jack had been studying sharks. Here's a photo of here's a photo of Jack Casey in 1964. When he discovered the nursery area for white sharks off the coast of northern New Jersey and Long Island. So Jack was very curious about sharks, and the federal government at the time was curious about sharks. And he began this research program, which included the cooperative tagging program. So he pioneered putting very small tags on sharks that were just marker tags. We call them conventional tags now. And what they do is they just have a little serial number on them. You put them on the shark. If the shark ever gets recaptured, you get 2 data points: where it was tagged, where it was recaptured, how far it traveled, maybe some growth rates. And he established a shark tagging program in 1964. He was offering this job at his laboratory, and I applied for it and quite excited about it. Working with a shark program, I was inspired by shark biology. I wanted to be a shark biologist, and I didn't get the job. That's fine. That's totally fine. I was kind of pissed off at the time. This was 1982. And I said, okay, how about if I just volunteer for the program? And I volunteered for the program in late 1982, and I was starting my last year at University of Rhode Island, and I decided for the last semester at the University of Rhode Island, I would go to the Caribbean and study coral reefs, because there were no jobs in shark science. We knew everything there was to know about sharks. Well, right before I took off, Jack offered me a position in his program as a technician. And that's really where I started to cut my teeth, so to speak, with shark research. So from 1983 to 87, I worked with Jack and his program. And what you see pictured here is me, the little punk on the far left side here, Chuck Stillwell, Jack Casey with the orange hat, Nancy Kohler, a fellow graduate student, George Benz, a very close personal friend. Not at the time, I had just met him, as you will learn in the book. And then Wes Pratt, who I worked directly for. So I was at this program and starting to do research cruises where I actually tagged my very first shark. This is a hammerhead off the side of a research vessel. We were setting all kinds of gear, longline gear specifically, to capture sharks, to tag them, to bring some on board, to dissect them. So it was during this 5-year period, being mentored by those scientists at the helm, Jack Casey at the helm, that I really started to better understand fisheries science and shark science and how little we knew about sharks. It's also with that program that I started diving with sharks. This is a shark cage that we built. Wes Pratt shaved off his beard, but he's the third from the left. I've still got my mustache, a second from the left. But if you look next to Wes Pratt, right in the middle, for those of you who might know a guy named Brian Scarry, that's him. That's Brian Scarry. In his early years, he used to do shark cage diving, but long before he was a National Geographic world famous photographer, and he was just cutting his teeth as well in that field. So I met some great folks, and we started diving in sharks. And what you'll see in the book is my early experiences and the way we clung to the cages. And over the course of a couple of years, we decided that staying in the cages was not all that important anymore. But you notice I'm not talking a lot about great white sharks, because what we knew about that species at the time was remarkably very little. It's an incredibly elusive species in those days. My very first white shark I saw while working for Jack and Wes in the program was in 1983. And this is actually the loading dock at Mystic Aquarium. And what you see, I'm a suspended. I became the photographer for the research program, Jack's program, as well as an histologist and a field biologist for them. And what's happening here is this white shark was harpooned by fishermen out of Noank, Connecticut that year, and it was brought into the docks. You'll if you read the book, you'll know exactly what you'll learn how it goes. Ultimately, it ends up in the back of Mystic Aquarium, where a bunch of taxidermists came and made a mold of it, and that's exactly what you see happening here. A mold is being made, and I'm suspended over the shark, documenting this. We later took what remained of the shark and did a necropsy on it. So a lot of our work in those days, and specifically my work, and this actually, led to my master's degree at University of Rhode Island, involved working with dead sharks. The technologies, that Frank Carey built in his lab were not being built commercially anywhere on Earth. And so he was the only one really doing that. And Jack was using conventional tags, which were very simple marker tags. And the more sophisticated technologies that exist today were simply not there. And so most of what we were doing was tagging with conventional tags and studying dead sharks. And we would do necropsies. So this is a really small white shark that was brought into the lab at that time with scientist Chuck Stillwell laying next to it. And this is a very large white shark that was brought in at that time. This is one of those specimens that was harpooned in the 80s. And so these specimens would come in and we would do necropsies and tissue sample, and we would learn about their natural history. So for example, the more obvious things you would do is look in the stomach of the shark and you find out what it eats, right? You look at its reproductive organs, male, female, mature, immature. If you look at its vertebrae, you can get a sense of its growth rates. So most of what we knew about white sharks related to dead specimens. In 1985, Wes Pratt and Jack Casey published a paper on the distribution of the white shark in the Western North Atlantic, and it was based on all these dead specimens that they could possibly find that had interacted with fisheries. And this told us basically the distribution of the animal at that time. We updated it in 2014 with one of my students, Toby Curtis, years later. But this is about 615 dead sharks that had been observed, caught by fishermen, brought in. And we felt that was the state of the art relative to what we knew about the distribution of the species. So what I want you to do is remember this image, because I'm going to show you how it's only partially right, So we sampled a lot of these animals. And like I said, if you take a section of the backbone, you can get a growth curve. So this is a paper we published not that long ago based on counting rings in the vertebrae of the shark. But again, most of this dealt with basic natural history. Very little had to do with behavior, migration, movement ecology. What do they do from week to week, day to day, year to year? Where do they go? How do they move through the three-dimensional environment of the ocean? We didn't know anything about that, And we weren't going to at that time because white sharks were so elusive. With only 600 points dating back to 1800, the chances of me raising any money to go out and say, I'm going to go study white sharks was really, really low, But my years at the Narragansett lab were fantastic and provided me with the tools I needed to move on. In 1987, I moved out to Martha's Vineyard Island. And this is the second major part of the book, where I kind of grew some independence and established in 1987 the Massachusetts Shark Research Program, which on paper really just looks like a smaller version of Jack Casey's National Marine Fisheries Service federal program. And it started with me being out there and working with local fishermen. And one of the great lessons I learned from Jack is he could walk down any dock and talk to anybody on that dock and learn something. You know, and John mentioned the fact that, yeah, we had this meeting and it was wonderful for him, but it was equally great for me because every conversation I have, whether it's one of you coming up to me later and telling me a story, or whether I'm talking to fishermen on the docks, I learn a little bit of something, you know, and the fishermen, whether it be recreational or commercial, you know, in those days, it was primarily recreational, but since then I've moved on to work with a lot of commercial fishermen, know a lot about the ocean and the animals that live with it. So from 1987 right up to 2009, you know, I established this shark research program and studied a lot of different species, not only locally, you know, from sand tigers that you see in the upper left to smooth dogfish. John and I did some smooth and spiny dogfish work together. That one's got an acoustic tag on it. Big world record mako shark. I'd sample at shark fishing tournaments. Again, continuing with the tissue sampling. My master's degree was on blue sharks. This is a poor beagle here. In the middle is the basking shark. We were doing basking shark work. And over that time period, new technologies emerged. You know, the acoustic tags that Frank Carey was building were being produced by commercial manufacturers and not built to last 3 days, 8 days, 10 days, but to last years. You know, satellite technology took off. The components of tags began to shrink. Sensors became more sophisticated. Batteries became more sophisticated. And as John and his staff will tell you, these are the tools we use today. So over the course of my career, it's been really fascinating to move from not having cars to having Corvettes. You know, that's a really cool time to have been a scientist. And my research not only stayed in New England, but, you know, I moved around the world as I collaborated with other scientists. I did whale sharks in the Red Sea. I did Greenland sharks under Arctic ice. I was in the Central Pacific doing gray reef shark stuff. But none of these are white sharks, right? And in every shark biologist's mind, somewhere tucked in there, they always want to see the Holy Grail. And the Holy Grail is the white shark, you know? And then 2004 came around, and who remembers this? You know, you do, you know, a white shark swam into a salt pond in the Elizabeth Islands, not far from where I lived on the vineyard, only a boat ride away. And this is a chapter, of course. Everything in this talk is a chapter in the book. And these were 2 beautiful weeks and painful weeks at the same time with this white shark that we tagged with a highly sophisticated satellite tag called a pop-up satellite tag, which we had been using on basking sharks. And I thought this was it. I thought this was the pinnacle of my career. I got a satellite tag on a white shark for the first time in the Atlantic Ocean, and I was going to learn so much information from it. And I'm always torn whether I should tell you how that chapter ends, because it's not as good as you think. And so I'm going to leave you hanging there. But this was an indication that, yes, Greg, don't forget, white sharks occur in New England, okay? And they have historically. Remember all those little green dots. But something was changing, and that's where we get into part 3 of the book, you know, as white sharks came back to New England in bigger numbers. And that's driven primarily by one species, at least off Cape Cod, and that's the gray seal. So in 1972, the United States passed the Marine Mammal Protection Act. And since then, marine mammals have been afforded the highest level of conservation. And as a result of that, going on in the background of many of our lives, unnoticed by many unless you work on or around the ocean, including me, seal populations began to rebound. A lot of marine mammal populations began to rebound. We were seeing conservation success. But when you reopen a restaurant, you're going to get the customers, and the customers are the men in the gray suits, right? And so with seals coming back to recolonize Cape Cod and the islands, sharks have taken notice. White sharks have taken notice, and white sharks have been afforded protection since 1997. And fewer white sharks were being killed, and there are indications through our data sets that indicate they're starting to rebound as a result of protection as well. And so you get this scenario that has now developed off of Cape Cod. So I'm working on Martha's Vineyard, trying to study sharks, hoping to study white sharks someday, and right in my backyard emerge great white sharks. I mean, that's just luck, you know? And Jack Casey taught me a great line. He said, sometimes, Greg, it's better to be lucky than good. I don't know if he's telling me something there. But I got lucky that was luck for me. And I had the tools, and I think I had the skill set, and I certainly have always surrounded myself with people who, quite frankly, are better than me. And I like that, because that's what it's all about. You know, being able to mentor people and bring people in that are great scientists, train them as best I can, and watch them take off is something I love to do. And I do it through UMass and other systems. But we were pretty set up in 2009 when I got a phone call from a spotter pilot. Now, for those of you who aren't familiar with spotter pilots, we actually have an industry in Massachusetts and New England where pilots go in the air, locate large fish like bluefin tuna and swordfish, report them to fishermen who are in boats who go out there, harpoon them and harvest them and sell them. That's been going on for decades. So we started using spotter pilots to locate bluefin tuna and basking sharks to tag. And I was working with a boat, captain by the name of Captain Billy Chapralis, on a boat called the Easy Does It. And we had tagged bluefin tuna while they were free swimming. And we had tagged basking sharks while they were free swimming. So we're not handling these animals using a modified harpoon technique that he developed. So again, a great example of partnering with people who know what they're doing on the ocean. And that's primarily, a lot of them are fishermen. And so this is 2009, one of the first white sharks we ever tagged. And it's from the pulpit of the fishing vessel Easy Does It, which was owned by Bill Chapralis with his son, Nick Chapralis, as a mate. And so in 2009, I got a phone call. I was at a really boring fisheries meeting, and Billy's spotter pilot said to me, it was just prior to Labor Day weekend, I think I see white sharks off Cape Cod. And I said, really? So I told my boss at the meeting, he let me go. And I drove down to Falmouth, hopped in this guy's plane. We went out and we saw 5 white sharks. And that really kicked it off, because the very next day, so I called Billy on his boat. We've been using this boat to tag those other species. He said, I'll move the boat around to Cape Cod. We'll go out and try to tag them. And so this is one of those first tags placed on a white shark in 2009. It was 5 pop-up satellite tags. And this to me was, very rewarding, because, and I talk a lot of, we talk a lot about it in the book as a major step to advancing what little we knew about the biology of the white shark in the Atlantic, not in terms of its natural history necessarily from tissues, but live sharks. So my career was transitioning, not 100% away from necropsies, because I still do them if the opportunities present themselves. If a white shark beaches itself and is dead, I'm going to learn from it. But transitioning to working with live animals using these newer technologies. And so in about this same time, anytime you study a big charismatic species, there's always some production company that's hovering over you saying, we want to film you. We want to show your work on TV and make you look like a star, right? And a lot of young scientists bite at this thinking this is going to be fantastic. And then they see the show and they go, oh God, how embarrassing. But I was really fortunate, because during my years in Narragansett, I met some filmmakers that were working with Jack Casey and Wes Pratt, and they became friends. And so when they called me, we started making shows together. And I had this level of trust where I knew there would be a show produced that had science and a science message and a conservation message reflecting what we knew and didn't know about this animal, where I hope I would perhaps inspire somebody to be a scientist, to learn more, and to respect these animals, if not, at the very least, and want to protect them. So in 2010, we were filming off Cape Cod, and this is the largest white shark I've ever tagged. This is Curly. Curly was feeding on a dead humpback whale 15 miles east of Chatham, Massachusetts. My spotter, I got a tip about a dead whale floating out there. I sent out the pilot. He saw a shark on it. We shot out there with the boat, camera, systems, tags, and I put a very sophisticated satellite tag that you see her towing right here. That tag is designed to come off after a year and report to me everything she did, while she was out. She was 18 feet long and probably in excess of 3,000 pounds. And to tag her was an incredible experience. And I thought after I tagged her, okay, we can watch her for a while, then we're going to go in. And the producer on board said, no, Greg, now you got to go in the water. And so I'm thinking, well, why do we have to do that? He goes, well, because it's part of the film. Okay, so we got in the water and I took this photo of this beautiful shark, and she was feeding on this whale. And to dive with a dead whale is not a pleasant experience, but to dive with a big, beautiful white shark is. But at one point, and again, Curly consumes a chapter in the book, Curly decides to eat one of the large orange polyballs holding the cage up. And I don't know how well, this is a dark photo. I took it from inside the cage. So this is my perspective from inside the cage. And next to me is Nick Kalionis, the cameraman that I've been working with for decades now, and a phenomenal filmmaker. And he's filming this whole thing. And she doesn't eat the big red ball, but she does get tangled up in the rope holding the cage to the surface and decides she wants to get free. Well, when a 3,000-pound animal decides it wants to free itself from something, it tends to want to smash that something to bits. And to be inside that something is not the most pleasant place to be. So this is all in the film, which is later named was Jaws Comes Home, one of the many dumb names that the films I make are called. But the content is good. Where she decides she wants to get free of the cage and she breaks it apart, and I'm in it. And I'll let you read the book in order to figure out what happened. But I'm here. I used to be tall, but I'm here. So Curly was and remains the largest white shark we ever tagged. She traveled to Florida for the winter. And because her tag is designed to come off, we really don't know where Curly is anymore. But we estimate her to probably be in excess of 50 years old by now. And part of our growth curve determined that these sharks live in excess of 70 years. So let's fast forward a bunch of years. Billy Chapralis, who's a commercial tuna fisherman, and I parted ways because we figured out an easier way to tag without using a big boat with a very slow boat. We moved over to a really small boat. Billy and I remained friends, but he went back to harpooning fish and I started tagging from the pulpit of the boat. And this boat was called the Aleutian Dream. And the Aleutian Dream is part of the Atlantic White Shark Conservancy. So a lot of my story on Cape Cod is associated with this nonprofit organization that was established by Cynthia Wigrin in 2012. She came to me one day and said, why aren't you doing more to tag white sharks? And it's actually a fun part of the book because it was somewhat of a contentious relationship to begin with. And I said, because I don't have the funds to do it. A lot of our problems early on in these early years from 2009 to 2014, 15, is we didn't have money. These tags were costing somewhere on the order of $4,000 each. And we just didn't have the funds. The state wasn't going to cough them up. Nobody had been hurt by sharks, so there was no reason to study them, right, at that time. And Cynthia stepped up and said, I'm going to start a nonprofit. And that's exactly what she did. And part of their mission is to support our research. And they have done that amazingly since 2012. So we started tagging from the Aleutian Dream using even a simpler technique where I wasn't throwing a harpoon. We were placing a small dart at the base of the dorsal fin, as you could see in this photograph. And as you can also see in this video that was shot by the Malibu artist who visited us from California to take this amazing footage of a white shark being tagged. Our approach is pretty straightforward. We use a spotter plane to locate the fish. Once he locates the fish, he radios the location, the general location, off Monomoy, off Chatham, off Orleans, various landmarks down to the boat. The captain on the boat is in the tower. He will run over to that area. I'm on the pulpit of the boat. I'm holding about a 12-foot long tagging pole with a small needle. and a dart, and the dart is to be inserted at the base of the dorsal fin. And so you're going to see me do that right now. We're not capturing the shark, handling the shark, pulling it out of the water, doing any of those things to it, because we're trying to study natural behavior, and I'm going to place that. You'll see me go down into the water right at the base of the dorsal fin. The shark's going to react because it's, you're walking up to you're going up to a shark and poking it. So it's going to react. It goes down, to the bottom typically, and then comes back up and behaves like a normal shark. So we're trying to be as least invasive as possible. And the tag I just put on that shark is an acoustic tag. The exact same technology, not exact, the same technology that Frank Carey built in his lab in 1979, but refined so that those tags last for up to a decade. So we can monitor the movements of these animals in three-dimensional space over big time scales, individuals over big time scales. But over the same period, what emerged on Cape Cod? The overlap of a top predator doing what it naturally does, and that's feeds on seals, with humans that became really, really accustomed to not having sharks around, right? As a matter of fact, they didn't even know there were supposed to be seals and sharks here, right? Because there hadn't been seals in over a century in any numbers. And so Cape Cod became a tourist-based economy, and people flocked out there to enjoy the beautiful beaches. So now we had this developing. So we talk a lot in the book about, human thinking and our approach from a scientific perspective, but also, public opinion, which ranged from, hordes of people with pitchforks and torches to the opposite end of the spectrum. And the complexity of the issue, what happens when you restore a top predator to an area. I mean, Ret writes eloquently about this in terms of wolves in Yellowstone, right? And it's a similar issue. We've become so accustomed to not having to worry about things in the water that might hurt us. And we've had now 5 incidents that have occurred off Cape Cod. 3 were actual bites on people. 1 was fatal. And so a lot of our research has evolved in recent years from moving away from big picture movements and to fine scale behavior. So as I wrap this up, I'm going to show you some of the latest and greatest technologies. And again, I can't say enough about how fascinating it's been to live through this period where we're not only attaching acoustic tags that last for 10 years, but we're putting camera systems on sharks. And that's what you're looking at right here. This is a camera system that measures what this shark, not only records what the shark is doing by riding the back of the shark, but also measures what the shark is doing 20 times per second in 3-dimensional space, turning right, turning left, accelerating, decelerating, depth, temperature. These are highly sophisticated things that I didn't have a laptop at URI, right? I used a big mainframe and with stupid languages called BASIC and Fortran. Who remembers that? Oh, good. There are other people in the room. I feel better about that. And now we're doing this, right? We're riding the backs of sharks to study their behavior. This is a shark we named Snoop. Snoop, this particular day, was swimming along the bottom off Cape Cod, snapping up and eating dogfish. We didn't know, I had a sense, we knew from stomach contents analysis that they eat dogfish, but now we're actually watching them do it, right? If I told you to go out and watch a white shark eat something, you will spend the next 50 years and never see that, right? Now we have the technologies that allow us to spy on these animals, and we don't even have to be there. I can sit in my office, put my feet up and pretend I'm working. So it's been a really, really exciting time. This shark is just gorging itself on schools of dogfish that occur off Cape Cod. And, I'd have to slow it down. What you're seeing there is, the shark actually eating the dog as it's crashing through those schools. That's what's fascinating. So I'll end with just these last couple of slides. We'll start with, Frank Carey's original track in 1979 and the new technologies and the team, which includes, one of my graduate students who is now finishing her PhD, works for the Atlantic White Shark Conservancy, Dr. Megan Winton, who's really the future of what we're doing. You know, just as the torch has been passed from Carrie and Casey down to the Skomals, it's moving on, you know, and I think that's what it's all about. And we've been able to really explode the science, respecting the pioneering work and turning it into that, you know. Those are the movements of white sharks in the Atlantic Ocean based on the new technologies. And we've done that over the last decade. And with that, before Ret comes up and reads a little bit more, I'm going to finish with this. I think this is a beautiful little video that one of the board members of the Conservancy made that shows our work and the beautiful area we get to work in and what a natural restored ecosystem really should look like, you know. But I didn't choose the music. That's my office. It's not too bad, huh? And the last thing I'm going to say is I'm going to thank my wife. It's her birthday today, and she was so nice to come with me, and she supported me for decades, puts up with me, and if you know me, that's a tall task. Thank you, Kimberly, for everything. And with that, I want Ret to come up.
Happy birthday, Kim. So I started with the beginning of the book. I'm going to end with the ending of the book. Greg filled in the middle, so guess you're done. No, please do. If you're interested, we do have copies of the book for sale out in the lobby. Every copy that's here tonight that you buy, my wife, who did the illustrations for the book, she has done a hand-drawn shark illustration on the title page. So it makes that kind of special if you're interested in that. The only thing you need to know about this section is that Ellis and McCosker wrote a book called White Shark in 1991 that was very pessimistic towards the future of the white shark. They were very concerned about the conservation status of the white shark at that time. When people advocate for an imperiled species, the situation may seem so desperate that it's hard to imagine what success might look like. It's even harder to imagine how that success could trigger a return to the very same mentality that caused the species to be imperiled in the first place. But that may be what we are seeing in the Northwest Atlantic with white sharks, a shift in perception. Restoring an apex predator to an ecosystem is a monumental task, and there are few successful examples. In part, this is because we, as humans, are loath to give up territory we've claimed. We return the gray wolf to Yellowstone, but we shoot it when it leaves the park boundaries and enters our territory. Since the second decade of the 20th century, we've claimed the ocean as our escape, our respite, and our playground. We slayed the monsters that inhabited it, and we tamed it with lifeguards and a bevy of amenities ranging from toilets to food huts. In our collective view, the ocean became safer, and over a century, we slowly altered our behavior toward it. We developed more seaside resorts and beaches for recreation. We spent more time in the water. We introduced our children to it. We learned to surf and boogie board and paddleboard. We designed wetsuits, allowing us to extend our range northward and longer into the colder months. Ultimately, we gave ourselves the impression that we had conquered it. We made it both suit and serve us. Now the white shark has returned to one of America's most iconic summertime destinations, and it's challenging our perception of what the ocean is to us. For the first time in a long time, we have a hazy sense of what it means not to be the top predator. For the first time in a long time, we've had to consider what it means to be prey, even if we're only mistaken as such. What do we do with these emotions? Do we celebrate our ecological success, the restoration of an apex predator to an ecosystem, or do we defend our hard-won territory? Is there a middle ground? We have some hard decisions to make, and how we make these decisions will say a lot about us and how we perceive our place on this planet. Ellis and McCosker struggled to find optimism at the end of their book. Extinction and endangerment are our legacy, they write, and included on the long list of the world's depleted species is the great white shark. There are many more reasons to be optimistic about the white shark today, but that optimism will only prevail if scientists like Jack Casey, Greg Skomal, and Megan Winton continue to carry the torch from one generation to the next, motivated by our admiration, respect, and even a bit of fear for these remarkable animals.
You have to
Check.
consume all 6 of those waters in the next 10 minutes.
After all that talking, I could.
All right. So we have about 10, 15 minutes. For the sake of time, I'm not going to ask my own questions. I have a million. The book is wonderful. You both should be incredibly proud. Ret, amazing writing. And happy birthday, Kim. But I'm going to take some questions from both the audience, which I will repeat for our virtual audience. So try to keep your questions succinct, because I'm not the best at remembering every little nook and cranny of what you're going to ask. So try to keep the context down, but be to the point. And then we'll take some questions from the virtual audience as well, which we're getting through the live feed. So why don't we start with a question from the audience? Yes, blue jacket. So the question is, can Greg explain the harpooning process he uses to tag the sharks in the wild?
All right. So the free swimming tagging process. So I walked through it a little bit up there, but plane locates the shark, relays it to the boat. Boat goes to the area. I'm on the pulp of the boat. I'm extended out, because if the boat senses, hears, feels the boat, it's going to go down, right? And so being out on, that's the same reason harpooners have pulpits on their boats. They sneak, it's a very effective way to sneak up on an animal. Then the tagging pole itself has a very thin needle with a small intramuscular dart. And the intramuscular dart technique was developed by Jack Casey in the 60s. And it's been used worldwide, you know, and what you do is you target the base of the dorsal fin. And one of the things you're trying to do is embed that dart into the supportive elements of the fin itself. All right. So it's an area that's chosen because it's not heavily vascularized, not a lot of blood vessels or nerves. And it's proven to be very effective. The goal being to minimize our handling and any invasive approach to the animal.
Thanks for the question. One from the virtual audience. Do you have an estimate for the population size off Cape Cod, white sharks? And do you think it's reached equilibrium?
It's a wonderful question. A couple of months ago, Megan Winton, as part of her PhD, completed the last chapter. And that chapter had to do with a population estimate for Cape Cod white sharks spanning the time period of 2015 to 2018. The number of white sharks that visited Cape Cod was 800, estimated over that period. I always tell people after I give that number out is they're not all there all the time. Okay. Doesn't mean there's 800 sharks swimming around Cape Cod, you know, as we speak. But that was the population estimate. You mentioned equilibrium, and I thought the population was increasing after 2018, but the last 2 years, I think it's starting to reach an equilibrium that's a little bit lower. Where it's reaching what we call perhaps the carrying capacity for that area. And I don't know what that number is, but based on our sampling, it appears that it's coming down a little bit.
Thanks for the question. Someone have a question for Ret, or else I'm going to sneak one in for him. All right, right there. So the question is, what was Ret up to in Wyoming? And did that have anything to do with the Yellowstone wolves that Greg referred to?
Yeah, that was actually one of the reasons I was so excited about this project. I did a bunch of writing. I work as a journalist, science writer, and I'd done a bunch of work about the wolf restoration in Yellowstone in the 90s. And there's so many similarities between what's going on with sharks in the Western North Atlantic and what was happening in Yellowstone. That was very compelling. I was doing a bunch of other stuff too, but wolves are a big part of it.
I'm going to sneak one more in for Ret, if I can. As you were writing this book, obviously Greg's story goes in so many really interesting directions, and there are so many potential directions to go in. And you did a wonderful job as Greg noted earlier, of weaving in the biology side, the conservation side with his story. What was the biggest challenge in kind of putting that together, besides having to, hang out with him?
And his technology. The biggest challenge, without a doubt, was the fact. So as a science writer, my happy place is being in the field with scientists, embedded with scientific teams in places like Papua New Guinea or the Solomon Islands, and reporting on what they're doing. But because we started this book as the pandemic began, we actually had our first meeting in March of 2020. And because Greg works for the state and there were very strict protocols that the state had in effect for a very long time, we had to do most of our heavy lifting early on via Zoom. And that was a huge challenge for me as a writer. And it's a huge credit to Greg. And again, as you all heard tonight, I mean, Greg, one of the great things about scientists like Greg is their ability to communicate. That's usually my job as a science writer, is to take these sort of complex ideas and give them to a general audience. Scientists like Greg make my work not nearly as important because they do such a good job of it themselves. But that was the amazing thing working with Greg, was just his ability and his willingness to open up and share his story with me. So even though we were doing it via Zoom, I think we still managed to get a very immediate.
I'll be honest, that it wasn't all that easy for me. There's always a battle between the logical side of me and the more illogical side of me. But Ret would ask me a question about, I'll tell a story about Curly, and he'll say, then follow it up with, well, how did that go? I say, it was great, it was cool. And he'll go, no, Greg, I need to know how you feel. And I go, feelings? I got to talk about feelings. I don't want to talk about feelings. What do you mean? No, Greg, that's what the book's about. You got to dig a little deeper here. And I was a little bit of a struggle for me. And for those who know me really well, that's not easy for me to do. So, but he pulled it out of me, I think. I think he did a great job.
Fantastic. Okay. Question from the audience. Yes. With blue striped shirt. Yes. So the question is for Greg: how many sharks does Greg see a year that he's tagging that are the same year over year?
Yeah. So one of the major parts of Megan's work was she established the white shark catalog. And if you go to atlanticwhiteshark.org, you can see that she's got over 600 white sharks in there. We could tell sharks apart based on their color patterns. So even if I haven't tagged it, we can later go back to, I videotape every single shark I approach. And so we can go back to the video and look to see if we already know it. And it's remarkable that we know a lot of them, not only because we hear the tags. Like the other day we were out, I saw 12 sharks last Thursday, and 11 of them were already tagged. Okay. So they do come back year after year after year. So they love the restaurant. Some of them get a hotel in Orleans, and and others just move on. But the struggle for me, actually, the last couple of years has been finding sharks that are not tagged or not in our catalog. There are a lot of repeat customers.
I'll just note that two prominent figures in Greg's arc, Dr. Wes Pratt, who he showed some terrific images of and is featured heavily in the book, and John Chisholm are both formal affiliates of our New England Aquarium's Anderson Cabot Center for Ocean Life. Can you maybe do a quick note, Greg, on John's role in helping to validate and identify sharks, either through Sharktivity or any of the other video ID he's done?
Yeah, I mean, I'd keep you people here way too long if I didn't cut this talk way back, but it includes photos of John Chisholm. John's been working with me since Gretel swam into the salt pond in 2004. He's a walking encyclopedia when it comes to sharks and shark literature, and absolutely amazing. He currently runs the Sharktivity app. So if somebody's sitting on a beach and sees a shark, takes a picture, sends it to Sharktivity, it doesn't go up immediately. It gets vetted by John Chisholm, and that's not easy to do. Particularly when you get a bunch of people who might be drinking beers on a beach and decide they want to send a photo that was in Time magazine in 1972 of a white shark. And he knows it. He goes, check this out, Greg. You know, this was the cover of Time, you know. It's like, so he's really amazing at doing that. And that's how we have confidence, and the aquarium's been instrumental in having John on board to do this. We have confidence in what's up on Sharktivity.
One more question from the audience and one more from the virtual audience. All right, who's the lucky in the white shirt? I'm not going to try to repeat that. Oh, so how do you find, Ret, the story around the research, I think, is the main point of the question.
Yeah, I mean, so Greg and I share the same agent, and it's really our agent, Susan Canavan, who put this project in motion and put us together. And when Susan originally talked to me about it, and then when I had my first conversation with Greg, to me as a writer, you know, I'm always looking for an arc. A story needs to begin somewhere, needs to go somewhere, and something needs to change. And that's really important. And if you don't have that change, you don't really have a story. And to me, it was Greg's remarkable story of being this kid who's enamored with white sharks because of Jaws, and then goes on to becoming a shark scientist in New England and realizing I'm never going to be able to study white sharks because I work in New England. And then all of a sudden to have white sharks show up on his doorstep. That trajectory is beautiful. But then we also have the story of the shark itself coming back. And so those two stories intertwine. And I think that's what makes the book really come together. And so, yeah, so it's all about arc, I would say, not to get too technical into the writing side of things.
Great question. Okay, so I'm going to do two lightning questions because I love both of these. That's okay. That means you have to keep your response to 15 seconds or less.
Yes.
From the virtual audience, this is from the UK. So thanks for tuning in. Are rising global water temperatures affecting the geographical boundaries of the species, meaning the white shark? I live in the UK, and we are seeing more and more sightings of species not common to our shores.
All right. The short answer is the historical distribution of the white shark has not changed. However, I think the timing of their migration is going to. So when they migrate north, how long they stay is going to change. But we're not seeing any evidence of white sharks in the UK yet.
Soon. From the virtual audience as well. Hi, I'm Alyssa, and I'm 13. And I was wondering if when you were analyzing the stomach contents of the sharks, did you find if there were any that were consuming ocean pollution and plastic?
It's a great question, and we did not. But I will tell you this: our sample size is really low. White sharks are a protected species, so they can't be harvested. So the days of actually getting some white sharks samples are over. All we do is get a chance to see ones that might have beached themselves, and it turns out they tend to be unhealthy sharks, and unhealthy sharks have empty stomachs. And so, but I have not found any plastics. I found mostly empty stomachs.
Gentlemen, thank you. And congratulations for an amazing accomplishment with this book. We're so grateful for you to have joined us tonight. I want to note once again that the Lowell Institute provides funds that support this lecture series that allow us to show it free of charge, or show them free of charge. So I want to thank them again. If you enjoyed this program and want to help support our conservation mission at the New England Aquarium, please consider giving to the aquarium at neaq.org. I'd like to invite the in-person audience, and thank you again for tuning in for those that are tuning in virtually. But for in-person, please join us in the lobby. Greg and Red will be signing copies of their book. You'll be able to purchase those copies. We're going to close this session in a second, and I just appreciate if you'd hang out in your seats for a minute and allow these two folks to enter into the lobby. Otherwise, what tends to happen is everybody swarms and then they never get out there. So we're just going to have you sit tight, and then you can meet them out there. And I'm sure they'll answer additional questions that you weren't able to ask during the program as they're doing their signings. We look forward to continuing our lecture series on December 7th, when we'll welcome shark scientist and educator Jasmine Graham, who will join us on screen, to screen, I'm sorry, a new episode of her PBS Terra series, Sharks Unknown. We hope you join us then. Thank you for coming out. Thank you for joining us virtually. Thank you, Greg and Ret. Fantastic night. Have a great night.
Thank you all. Thank you all for coming out tonight. Thank you, John.